Hello everyone and welcome to today's podcast, which is about a brief discussion of scientific research paper. I am Saira from Knowledge and Research Support Services Department of University of Management and Technology. Today we have a special guest, a lead author of the fascinating research paper published in our journal. Welcome to the podcast. Please introduce yourself.
Okay. I'm Dr. Ayesha and I'm currently serving as assistant professor at the Institute of Microbiology and Molecular Genetics, University of the Punjab.
First of all, thank you so much for the availability and willingness to share your experience regarding your research you published in BioScientific Review in the volume 6, issue 3, 2024, entitled Bacterial-Assisted Phytoremediation of Selenite in Mung bean plants. I want to share that the journal editorial board has decided to select 2 papers from the bulk based on clarity, novelty and importance of the research area, and your paper is one of those 2 selected papers. Congratulations.
Thank you, I'm obliged.
Considering the importance of your work, I would like to know what motivated you to explore the bacterial-assisted phytoremediation in Vigna radiata. Was there any gap in the existing research that inspired you for the conduction of this study?
My motivation for this research and experiments, they stemmed from the growing need to find the sustainable solutions to address the soil contamination, particularly contamination of soil with heavy metals. So phytoremediation is a promising technology, and often it is limited by the plant's tolerance or inability of the plants to tolerate the high concentration of contaminants in the soil. There is already data available that supports the bacterial-assisted phytoremediation as well as the biofertilizers or the use of the biofertilizers to support the plant growth. But there was a little lack in the comprehensive study or experiment where they have studied the synergistic effects of Vigna radiata, selenite and selenium, as well as the use of the biofertilizers to address the contamination problem. So this all inspired me to see whether or how much or how could be these beneficial bacteria could be used in the bioremediation setups.
Your study revealed the growth improvements in mung bean plants treated with selenium-resistant bacteria. Could you elaborate on the practical applications of these findings, particularly in agricultural and environmental contexts?
Okay, the promising growth promotion that was observed in the study or experiment, it suggests the practical application of these biofertilizers as well as the plants in agriculture, real world agricultural as well as environmental management. For example, in the agriculture, these bacterial strains or isolates, they can be used as the biofertilizers, which mean they can benefit the farmers to increase and maintain their productivity in otherwise compromised soil and regions of the areas. As well as environmentally, it does not only aid in the growth promotion or increase in the yield of the crops and the growth of the plants, it also aids in the phytoremediation, which means simultaneously they do not promote the plant growth, but they also bioremediate or phytoremediate the contaminants like heavy metals or any other contaminant that is present in the soil. So this has a promising application, and it it does not only address the problem of the crop growth or decrease in the crop growth or increase in the soil fertility, but also addresses the phytoremediation at the regions or the areas or the soil where the contamination of the other heavy metals and other pollutants and contaminants are high.
You found that the bacterial strains used were able to reduce not just selenium but other heavy metals too. What do you believe is the broader potential of these strains in multi-metal contaminated environments?
Okay, ability of these bacterial isolates or biofertilizers to not only biodetoxify or biotransform the selenite, but also other heavy metals at the same time, it suggests it has a broader application in multi-contaminated regions or soils or the area. For example, in multi-metal contaminated soil where the traditional remediation techniques only cater or tackle one contaminant at a time, these biofertilizers or the selenite reducing or selenite resistant bacteria, they can tackle or can biodetoxify or biotransform, whatever you call it. They can do this remediation or bioremediation of multiple heavy metals at the single time, simultaneously, and this suggests that we can use these biofertilizers or the bacterial isolates at the broader scale, and we can broader even the horizon of this research by going with the other plants or the crops as well, so expanding through the ecosystems and the multiple environmental areas.
Okay, Dr. Ayesha, what were the main challenges you encountered in isolating and characterizing the selenium-resistant bacterial strains, and how did you overcome them?
When you are dealing with the living systems or the biological systems, it comes with the optimization of the experiments. But as far as this experiment is concerned, the main concern was to isolate or select the bacterial isolates that do not has the capacity to remediate the selenite into reducing into elemental selenium, as well as they must have the ability to promote the plant growth. So various sort of media were used to select the strains that had the capacity to tolerate and resist the high concentration of selenite into selenium, as well as meantime they were resistant to multiple heavy metals, and they had the ability to promote the plant growth.
Can you explain how you selected the selenium resistant bacteria from the contaminated soils, and why the choice of Wigner radiator was critical to the success of phytoremediation studies?
Okay. The selenium resistant bacteria, they were selected from contaminated soil from industrial zones, and while they were isolated, they were selected on the basis of their phytoremediation abilities, their growth promotion of the plants as well. So as far as the choice of the plant or the crop is concerned, Wigner radiata, it grows well. It has rapid growth. It has extensive root system, it can grow in various types of the soil, and it is widely used and consumed legume, so which makes this crop an ideal candidate for the phytoremediation projects or the setups where they do not have the only capacity to address the contamination problem in the soil, as well as it can increase or improve the growth promotion or the yield of the crops as well. So it will benefit the global food security system in the affected regions.
Well, selenium seems to have played a dual role as both a stressor and a growth enhancer according to your study. So how do you believe a real world balance in agricultural applications?
In real world application, using selenium as a stressor as well as the growth enhancer, it requires careful monitoring of soil monitoring, as well as the regular targeted introduction or bioaugmentation of the biofertilizers. For example, in moderate concentrations or moderate levels of the selenium in soil, it enhances the growth, it improves the growth, it increases the yield, it enhances the stress tolerance of the plants, and it enhances the antioxidant activity of the plants. But as the threshold level is crossed in the soil, the various forms or speciation of the selenium can affect or they are toxic to the various forms of life, not only to the plants but to the other various forms of the lives. So here comes into play of the selenite reducing bacteria. when the quantity of or the levels of the selenium are increased in the soil, you introduce the selenite reducing bacteria, and they simultaneously not only promote the growth of the crops, but they are also address the problem of contamination, which means they will detoxify the selenite into selenium. So it has a broader application. We can use these biofertilizers with the crops as well into the various forms of the contamination and medium, and specifically in industrial zone where the contamination of the soil is increased with the multiple sorts of heavy metals. We can use these biofertilizers to address the problem of contamination.
Okay, I would like to know, are there any follow-up studies or collaborations you are considering on this research for the future?
Yes, I'm currently working on a few follow-up studies of this research. For example, one of my main interests to isolate the other biofertilizers as well, and to see whether these biofertilizers or other isolated biofertilizers work well with other crops or not. And then I am currently using other heavy metals as well, and the collaborations with soil scientists and environmental engineers are also in pipeline.
Lastly, Dr. Ayesha, I would like to know that being an author of the selected Editor's Choice article, share your experience in publishing with BioScientific Review, and how did you find the peer review process and submission process and all that?
My experience is pretty positive. The submission process was straightforward, and I appreciate the journal's commitment to transparency and rigorous peer review. Thank you.
Thank you so much, Dr. Ayesha, for your valuable time
so
and the information which Dr. Ayesha has provided us. I hope this information, this scientific information, is going to be beneficial for the future researchers, for the academia. Thank you.